Hi, everybody. I just wanted to go ahead and kind of go through some of the slides that we had. I've updated our affective neuroscience slides so that we actually have kind of a tighter and more up-to-date list of topics. So I took out some of the schizophrenic slides that we had before and cleaned them up a little bit, and also talked about anxiety disorders and a couple of things that we have here. So I've cleaned out some slides here so you can kind of, it flows a lot more closely to what we had in the classroom. And we talked about how anxiety disorders and affective disorders, this is correctly written as affective disorders up here. In the previous version you had, it was anxiety. And how these mood disorders can actually be linked to some aspects of our the stress and to the stress hypothesis in terms of HPA system and tactile stimulation and these types of things that are important for being able to have function occur. And the monoamine hypothesis I didn't talk that much about, but basically is one that we've talked about before in neurochemistry, where inhibition and breakdown of norepinephrine and serotonin seem to be really important in terms of being able to help promote and elevate mood. And so those are illustrated here in terms of their major kind of substrates that you have, and tricyclics inhibiting reuptake of both of these different types of systems. SSRIs here, like Prozac or fluoxetine here, is being able to inhibit it. Monoamine oxidase here is helping to inhibit their breakdown. And so that stuff's not really that surprising given what we've had before, but I just wanted to kind of tie that up a little bit and move then on to schizophrenia. So in schizophrenia, what we're looking at is basically a severe mental disorder and some symptoms with it. In the original slides, I had some descriptions that were sort of out of date, so I've basically kind of took those off. And what we're looking at basically in schizophrenia is a couple of different things, positive and negative symptoms that are associated with it. The positive symptoms include delusions of grandeur and paranoia, or delusions of persecution, disorganized thinking, and there's also negative symptoms, inability to think flexibly about certain types of things, and some types of disorders in terms of being able to sort of sort out whether something's actually important or not important. And we'll emphasize that piece of it a little bit more in a little bit. But in terms of schizophrenia and the types of things that are part of it, the components that are the biological bases of it is basically that there's some evidence through twin studies and even some types of genetic studies that they've found that are seem to correlate with schizophrenia. And so that's one type of thing that we have here is a genetic disorder. There's a gene called DISC1 that seems to have some aberrations in terms of how the neurons within the cortex actually wire in patients, and then they've modeled that in some types of stem cell populations that they've differentiated into neurons. And so they've also looked at some things and found like, okay, well, there's some cell loss there. Ventricles seem to be enlarged in schizophrenic patients. And there's other structural types of deficits that have occurred there, including some damage in the hippocampus in schizophrenia and schizophrenics. And so these types of things are, some components of it. There's also the dopamine hypothesis and the glutamatergic hypothesis. The dopamine hypothesis is basically with amphetamine psychosis, it's very difficult to differentiate somebody who has amphetamine psychosis who may not be necessarily schizophrenic and somebody who's having a schizophrenic/psychotic episode. And the treatment of this is basically like, oh, well, things that can actually help suppress dopamine signaling can be helpful in terms of being able to improve the symptoms and regulate the symptoms and positive and negative symptoms in some cases of schizophrenics. So there is also the glutamate hypothesis, and patients with PCP or patients that who've been admitted to the ER with PCP psychosis, and they inhibit NMDA receptors, and so this is kind of an interesting thing where we sort of shut down this glutamatergic type of signaling. And glutamate's an excitatory neurotransmitter, and one that's important there. So these two types of neurochemical types of bases for schizophrenia, what I wanted to do is basically try to bring us a little more up to date into thinking about how schizophrenia works and some of the mechanisms behind it. And so I've introduced a couple of different slides here. One is basically trying to emphasize what I would have normally talked about in class or drawn on the board is this kind of idea of sensory gating. So normally what we have basically is auditory or visual stimuli come in, and then they they are filtered. We filter out certain amounts of it. In schizophrenics, it seems that this is actually disrupted. And you can see this even in really basic and sort of core types of functioning here, where you actually give like a click, an auditory click here, and there's a response of about 50 milliseconds that actually of an EEG wave that we have normally to it. And then if we repeat the click again, another 50 milliseconds later, it's not as strong as it was before. And so this P50, named after that 50 millisecond time period, is an indicator that we've actually sort of attended or habituated slash maybe we're gated out that type of stimulus that we've actually been exposed to. And this doesn't happen as readily in the schizophrenic patients. And so this is an idea of like what they call sort of a failure to actually gate out that stimulus so that we aren't processing it. So in this type of very like 50 millisecond period, it's only a couple of synapses, right? About 10 milliseconds per synapse for what you're actually being able to process here. And so it's really a very core kind of processing that's occurred in these patients, and that's been affected. So, what's the rationale behind this and how can we bring some of these things together? Well, one of the things that they've noticed in schizophrenics basically is that if you compare it to normal subjects over here and you give them a complex task to be able to sort out, schizophrenic patients seem to have a decrease in the prefrontal, dorsolateral prefrontal cortex. And that's one area in these sort of complex card sorting tasks that they typically have. And there's other types of tests that they're actually using to do that. And so there's what they call hypofrontality is the idea of it, lack of activity here. And this is also sort of fits in with some of the idea of the glutamate hypothesis, where maybe there's glutamatergic type of feedback or some kind of disruption that's going on that could help explain what's happening in this. And so they've looked at several different types of ways that could fit together, and there's two hypotheses that fit into it. One focuses a lot on this prefrontal cortex. And then another one I'm going to tell you focuses on the hippocampus and digs into that piece of it. And I think the two can ultimately complement each other and help give insights into how the physiology of this type of thing works, and even how it may connect to some of the things we're thinking about with sensory motor gating. So let's dig in. And what we see here is that just a simple map of basically the synapse or the circuitry that we can see associated with this first hypothesis for the underpinnings of schizophrenia. So with this case, normally what they're saying is that this frontal cortex, dorsolateral prefrontal cortex like here, has some activity here that then these are glutamatergic neurons here, and that they positively link to this GABAergic neuron here shown in purple, and that helps to shut down feedback to this area here. This is the ventral tegmental area. These are dopamine neurons that are responsible for projecting out to either the nucleus accumbens, and this is responsible for a lot of reward types of sensitivity, and it's sort of the neural substrate for cocaine abuse and other types of things. And then what we also have is that the ventral tegmental area can project out to the cortex. And so there's kind of the mesolimbic and the mesocortical types of pathways from the ventral tegmental area. This is distinct from what we learned about for the substantia nigra, which goes to the striatum and is important in Parkinson's disease. But nonetheless, these are dopaminergic neurons, and they're important for functioning. So what I've basically said is I've described this kind of circuit, feedback here, and I've been a little vague about that because this feedback actually has two different parts to it. There's a sort of positive feedback loop here that goes to the nucleus accumbens. And so the idea is like, okay, you have activity here, and then that kind of shuts things down so that you don't necessarily have as much feedback going to the nucleus accumbens. And what that would mean is sort of that it decreases the saliency of a particular type of stimuli that you have here. And in that case, you're sort of gating out and being able to help control like what it is that you're getting. So something's not as salient as it used to be. So your prefrontal cortex is able to help kind of dampen and control that type of thing. And the other thing then is that you also have another branch of this that's important for being able to feedback to the cortical part of this. And in this case, what they do is that you have this sends its input down to an inhibitory neuron here that then shuts down the ventral tegmental area. So when this is active, then this suppresses the activity here. And what that means then is that when this is active, then we have less activity here, and so we have less saliency here, and then more ultimately more driving of this kind of functioning here, because what we've done is that we've been able to kind of drive this kind of dopamine function through the action. Right, so we go from the frontal cortex here, this shuts down here, which breaks the ventral tegmental area. So go through this kind of thing and make sure this makes sense to you. It's sort of like what we learned about for the Parkinson's disease, where we have sort of two inhibitory components in a pathway. And it should fit and it should make sense. This is one thing I want you guys to tune into. And the reason is the following. In the case of schizophrenics, what they think is that basically you knock this out, you lose the frontality piece of it for whatever reason. Some of it they think is GABAergic neurons get disrupted, and others have other different types of hypotheses. But there's something going on here that's lost. And so then what happens is that this pathway gets overactive, and you have saliency attributed to things that normally aren't. And then you also have this pathway that gets overactive that basically then shuts down this feedback that goes up to the frontal cortex. And so you have less kind of activation of the cortex here, ultimately leading to this hypofrontal type of, or perpetuating this hypofrontal type of scenario. So that's one kind of idea, and that's out there about how this potential system works. The other one that I wanted to go through is talk about the hippocampus. And I alluded to that before, but basically what the idea is that this hippocampus is really important for being able to help control the nucleus accumbens here, and the ventral pallidum here, which is part of the striatum. And then that goes to the ventral tegmental area. And so we can see that this flow chart fits up with the other one from another publication. Here's the VTA, here's the nucleus accumbens, and then the hippocampus is not depicted there. But we can see how some of these players connect. And so the VTA actually does talk to the nucleus accumbens. But we're focusing on this part right here. And what I like about this story, and what I think is really, a worthwhile exercise for us to do, is that it takes something from being a circuit into thinking about how the physiology and activity of the neurons change in different types of situations. So here's what people have found when they've looked at this. The hippocampus has glutamatergic outputs to the nucleus accumbens. The nucleus accumbens then suppresses the ventral pallidum area, which then in turn sends GABAergic neurons to the ventral tegmental area. So we think about this ventral pallidum and how it actually connects. What's going on here? When it synapses onto the VTA, it shuts down or silences those neurons here. And those neurons can be tonically active. Like here's an example of a tonically active one where it's firing and has a modest amount of activity in the background. And the ventral pallidum kind of helps to keep them quiet. And that's a benefit because then we can recruit those silent neurons and have them become more active if we need to. The VTA is one that's really important for helping to attribute saliency. As we talked about, the VTA talks to the nucleus accumbens. Remember, this thing from here to here, and helps to attribute saliency to a lot of different types of things. So having it quiet makes it so that we can actually have some kind of ability to sort of gauge whether something's super salient or not. We also have this input from this particular pontine area. I have to just double check and make sure it's it. Pedunculopontine tegmentum area here, PPTG. I always stumble over that one. But these are a cluster of nuclei that are also in the brainstem, kind of near the locus coeruleus and the raphe nucleus in the brainstem. But basically the idea is that they come up and they're excitatory into the ventral tegmental area and kind of juice it a little bit for saliency of a different type of stimulus. So if something's kind of a salient stimulus, it's going to activate this, and that in turn then helps the VTA talk to the nucleus accumbens and tell the nucleus accumbens, hey, put out a little bit of reward because this is something that's actually something we want to pay attention to. So in this case, when we have something that's really active here, we change the way that this fires, so it's a lot more phasic burst type of firing that ends up occurring from these neurons. So the thing that's kind of cool here is just being able to appreciate how we have this sort of inhibition period and inhibition of some of these neurons here, that we also have sort of tonically active dopamine neurons here in the ventral tegmental area, and then that we have this other type of input that's helpful for processing saliency that basically is helpful in terms of this active, phasic burst firing that describe it. So how does this stuff play out? And so what I want us to be able to do, or like you guys to be able to do, is to be able to kind of approach and think about how this works in schizophrenia. And so that's outlined in the following type of context. What we just went through now was just in the normal situation here. This is a simplified version of it where they're basically talking about, and they're focusing on the ventral palladiums and GABAergic output that's basically keeping some of these quiet here. And so you have some reserve for neurons to be able to fire if they need to or be recruited for activity. And then you've got some that are just sort of tonically active. And then you have the PPTG that's describing salience. Great. So a benign context, basically the PPTG is not as active as it normally is. So you don't have as many firing because there's really not, it's not necessarily a process that you have to worry about. And so the dominant influence then is this inhibitory type of input that comes in via the hippocampus all the way through to the ventral pallidum. So this inhibitory kind of influence is kind of dominant in that kind of case. In a threatening kind of context, the salience of the stimulus basically kicks up the activity of the PPTG, and that in turn then activates the VTA, and we get more of these engaged in this burst firing type of pattern. And so you have a dynamic range basically between something being benign or whether it's threatening here in the case of, when you're trying to determine what to think about a particular type of stimulus. And so accordingly, there's less dominance of this inhibitory type of influence from the hippocampus. So, okay, this is how it's working for us. What's going on in the schizophrenic case? Because there's damage that they're seeing in the ventral hippocampus, and or at least there's loss of sort of inhibitory types of components in the hippocampus. What seems to be happening is that this whole process seems to be shut down. And so what ends up occurring is that you have a loss of this kind of activity here, and instead you're finding that there's this dominance of this kind of activity that we have here for the PPTG, right? So now we have this problem of these being overly active. So that's a major thing that I wanted you guys to focus on. The next slide goes in and talks about how these different regions actually relate to cognitive and negative symptoms. I'm not going to ask as much about that, but it's really interesting. The ventral striatum is basically just talking about the ventral pallidum, or the nucleus accumbens, and then here's the ventral pallidum. This is a VTA here, and sort of the pathway that we were talking about for positive symptoms. And then these are linking some other regions for the cognitive and the negative symptoms here. And then finally, the last slide I wanted to kind of point out to you was just, it's kind of a zoom in on the limbic hippocampus and talking about how these GABAergic neurons get disrupted. And they affect this ability of these neurons to function. So that's the story there. Hope that was helpful. And it was, I guess it was a little longer than 5 minutes, but it was definitely, I think, worth it. And I kind of told you where to focus. So thanks. Bye.